question number 46. Question number 46 says that what is the role of NAD plus in the cellular respiration process? It functions as an enzyme, it functions as an electron carrier, it is the final electron acceptor for anaerobic respiration, and it is a nucleotide source for the ATP synthesis. Now, this question has been asked from the chapter of your respiration in plants, and again, we all know that this NAD plus, which it is asking about here, NAD plus is basically a reducing agent that acts as an electron carrier. This acts as a reducing agent acting as an electron carrier in the process of cellular respiration, because of which the electron cannot be passed on from one complex to the another complexes, or from one substrate to the another substrate in a chemical reaction. So this is again a very direct question that has been asked. So we can say that in cellular respiration process, the NAD plus acts as an electron carrier. So definitely it is not an enzyme, because if it would have been an NADP reductase, then it would have been an enzyme. All right. So option B is the correct answer. It is the final electron acceptor for anaerobic respiration. Again, no, definitely not involved in the anaerobic respiration, not the final electron acceptor. And it is a nucleotide source for ATP synthesis. Again, no. All right. So that's why the correct answer is that it acts as an electron carrier in the process. Now, question number 47. So question number 47 says that which one of the following plants shows a very close relationship with a species of moth where none of the two can complete its life cycle without the other? All right. So is it hydrilla or yucca or viola or banana? Now, this question has been asked from the chapter of your sexual reproduction in flowering plants, and that from the category of pollination of your standard 12. All right. And let me tell you that this example of moth has been given, and the plant which is involved in its relationship is known as the yucca plant. So yucca and moth, they are considered to be, or they are known to have a mutual obligate association with one another. So yucca and moth are known to have obligate mutual association with one another. Now, how does they exactly benefit it? How the moth benefits the yucca plant, or the yucca plant benefits the moth? So basically, this moth species is responsible for pollinating the yucca plant. So moth pollinates the yucca plant. And on the other hand, this yucca plant provides a safe side for oviposition, for laying the eggs to the ovary locules, in the locules of the ovary of the yucca plant. So in return, you can say the yucca plant provides a safe side for laying eggs. Which is also known as oviposition. in the locules of ovary of yucca plant. And as soon as the fertilization takes place and the seed starts developing out or coming out, the larvae of the moth that also starts coming out of the yucca plant. So this is how they benefit. They show obligate mutualistic association with one another. That means yucca and moth. So the moth is going to pollinate the yucca plant, and in return, the yucca plant provides a safe site for laying eggs in the locules of the ovary of, yucca plant. And here, basically, the moth species which is involved is Pronuba. All right. So that's what the correct answer is. Option B over here. Now, question number 48. Question number 48 says that oxygen is not produced during photosynthesis by green sulfur bacteria, nostoc, Cara, and Cycus. All these options have been given. Now, let me tell you that this question has been asked from the chapter of your photosynthesis in higher plants. All right. And this is a very primitive question that has been asked from the preliminary case. And let me tell you that out of all the categories of species that has been mentioned over here, like for example, if I tell you, then green sulfur bacteria is considered to be one such category of bacteria that doesn't sources, see, for producing oxygen. All right. So oxygen, we all know that during photosynthesis, we all know that the oxygen is mainly produced via photolysis of water. O2 is produced by photolysis of water. So it is basically produced due to photolysis of water. Like for example, if I just draw the, so in presence of light, and calcium ions and along with manganese, this water is going to split up into 4H plus oxygen plus 4 electron minus. So this is exactly the photolysis of water. So this reaction is the photolysis of water. All right, and here the oxygen is liberated, as you can see over here. All right, so in all these categories, whether it's nostoc, Cara, and Cycus, nostoc is a category of BGA, Cara is a green algae, and Cycus is a gymnospermic plant species. So in all these categories, there is photolysis of water, and that ultimately results into producing oxygen. But in case of the green sulfur bacteria, the oxygen is not produced, during the photosynthesis, because this for producing oxygen, there has to be the source of water. And in case of the green sulfur bacteria, they do not use water as the source of proton. Therefore, they do not evolve oxygen. So we can say that for green sulfur bacteria, they do not use water as source of proton. So since they do not use water as the source of proton, so therefore they won't be evolving the oxygen as well. Therefore, they do not evolve oxygen also. All right. So that's why the correct answer is option A. Now, question number 49. Now, question number 49 says that in which of the following forms in iron is iron absorbed by the plant body? All right. So iron is basically, again, according to NCRT, if you just see to it, and this question has been asked from the chapter of your mineral nutrition of your standard 11th, and this is a very direct question, because in the sections of your macro element and micro element, you might have studied it as well. So iron is basically absorbed. See, iron has two forms. Let me tell you that iron exists in nature in two forms. So it has two forms. So the one is known as the ferrous form, which is Fe2 plus, and the other is known as the ferric form, which is called Fe3 plus. All right. So according to the NCRT, if you will see, then iron is absorbed by the plant body in the form of ferric ions. And later on, it might get converted into ferrous ions. It might be available in the form of ferrous ions. So basically, if you just consider, then plants absorbs iron. See, plants preferably absorbs iron in the form of ferrous ion. But according to NCRT, if you are going to see to it, all right. So it has been written that iron is absorbed by the plant body in the form of ferric ions. So iron is absorbed by plant body in form of ferric ions. And this is according to NCRT I'm talking about. This has been written in your NCRT. But generally, if you will see to it now, then plants are going to absorb iron in both the forms. That means ferrous and ferric. And since you have to go with the NCRT part, so that's why we can say that the correct answer is option A. All right. Now, question number 50. The question number 50 says that which of the, question number 50 says that double fertilization is fusion of two male gametes of a pollen tube with two different eggs, fusion of one male gamete and two polar nuclei, syngamy and triple fusion, fusion of two male gametes with one egg. Now, this question has been asked from the chapter of your sexual reproduction in flowering plants of 12th standard. Now, let me tell you regarding the double fertilization. So, double fertilization is called so because it involves two types of unions. So, that's why it is considered to be as double fertilization. Now, out of the two male gametes which are produced or released by the pollen tube, so out of two male gametes released by pollen tube, all right, and this occurs in the embryo sac, this we all know, all right. So out of the two male gametes which is being released by the pollen tube, the first male gamete fuses with egg cell. So the first male gamete is going to fuse with egg cell of embryo sac. To form zygote, which is diploid. So here we know that the male gamete is haploid and the egg cell is also haploid. So they will fuse to form zygote, and this process is considered to be as syngamy. So process known as syngamy. Clear? So that is the case. Now, the second male gamete, which is left, the second male gamete fuses with diploid secondary nucleus. So this is going to fuse with diploid secondary nucleus, which is also known as central cell. All right. To form triploid primary endosperm nucleus. So to form triploid primary endosperm nucleus. So this process is considered to be as triple fusion. Why? Because there is involvement of 3 nucleus. So here 1 and here it is 2. So process known as triple fusion. So it is because the 2 types of fusion, since 2 types of fusions are involved over here, that's why this process is considered to be as double fertilization. All right. So let us take a look at the options that has been given. Fusion of 2 male gamete of a pollen tube with 2 different eggs? No. Fusion of 1 male gamete with 2 conal nuclei? No. Syngamy and triple fusion? Correct. Fusion of 2 male gamete with 1 egg? No. So that means option C is the correct answer. All right. Now, question number 51. Now, question number 51 says that which of the following elements is responsible for maintaining turgor in the cells? All right. So let me tell you that turgor pressure is that pressure which is exerted by the cell cytoplasm on its cell wall. All right. So turgor, or you can consider it to be as the turgor pressure. So the pressure exerted by cell cytoplasm on its cell wall. That is called turgor pressure. And for maintaining this turgor pressure, the one important ion which is very, very important is the potassium. There are basically 2 important ions which are very crucial, sodium and potassium. But out of sodium and potassium, it is the potassium that maintains the turgidity in the cells. All right. So this question has been asked from the chapter of your mineral nutrition, from your this one, 11th standard. All right. So turgor pressure is maintained by potassium ions. All right. So option D is the correct answer. Now, question number 52. Now, question number 52 says that pollen grains can be stored for several years in liquid nitrogen, having a temperature of. All right. So this question has been asked from the chapter of your sexual reproduction in flowering plants and in the pollen grain section of your 12th standard. So let me tell you that the process by which the pollen grains are preserved in liquid nitrogen, that process is considered to be as cryopreservation. And cryopreservation is done so that it's one of the important ways by which the pollen grains can be preserved for future reference or for future use, you can say. All right. So pollen grains are stored in liquid nitrogen. And let me tell you that the liquid nitrogen, they have a temperature of minus 196 degree Celsius for several years so that they can be used for crop improvement programs. So in the future, they can be used for various kinds of crop improvement programs. So option D is the correct answer. All right. Now the method by which the storing method is called as cryopreservation. So this method is called cryopreservation. This method is considered to be as cryopreservation. That means storing any living tissue at this temperature of liquid nitrogen is called cryopreservation. All right. Now, next is question number 53. Question number 53 says that which among the following is not a prokaryote? All right. So it is, this question has been, this is a generalized question that has been asked. We cannot say from a particular chapter, but this is a very general kind of a question that has been asked. All right. So the options include Saccharomyces, Mycobacterium, Oscillatoria, and then Nostoc. Now let me tell you that Saccharomyces, which is also considered to be as yeast. All right. And this is a category of fungus. This is a fungal species or a fungus. And we know that fungus are multicellular eukaryotic organisms. So these are multicellular eukaryotic organisms. So they cannot be included in case of the prokaryotes. And overall, generally, if we are going to see it, then this is a feature of multicellularity, is a feature of all the fungus. But yeast, if you are going to consider it, so this Saccharomyces is specifically unicellular. This is the only fungus which is unicellular. So you can say only unicellular fungus. Only unicellular fungus. So this is usually eukaryotic. All right. They are not prokaryotic, while the other options that has been given, Mycobacterium, Oscillatoria, and Nostoc. So Mycobacterium, Oscillatoria, and Nostoc, all are the categories. Oscillatoria and Nostoc, these are the categories of your cyanobacteria, while Mycobacterium is a bacterial species. So Mycobacterium is a bacterium species, and this is prokaryote. While Oscillatoria and Nostoc, if you consider it, so both these are the category of cyanobacteria. And we all know this is also a prokaryotic category, cyanobacteria. So that's why the correct answer is not a prokaryote, is Saccharomyces cerevisiae. Now, the next is question number 54. So the question number 54 says that the two functional group characteristics of sugars are, all right. So there are two functionality or functional groups that are found in case of sugars. So what are those functional groups? Hydroxyl and methyl, carbonyl and methyl, carbonyl and hydroxyl, and carbonyl and phosphate. Now this question has been asked from a chapter of biomolecules of standard 11. And let me tell you that sugar basically denotes carbohydrate. We all know this. All right. So sugar denotes carbohydrate. All right. So sugar is responsible for denoting carbohydrate. And carbohydrates, they are considered to be as the polyhydroxy aldehydes and ketones. They are considered to be as the polyhydroxy aldehydes and ketones. So carbohydrates are polyhydroxy aldehydes So they are considered to be as polyhydroxy aldehydes and ketones. So if they are polyhydroxyaldehyde and ketone, so they will definitely be having carbonyl group and the hydroxyl group. And that's why they contain, that's why they have carbonyl and hydroxyl group. Because ketone is basically denoted by, just a second. These ketones, they are basically denoted by like this. So this is ketone. And aldehydes are denoted by CH. That's all. So definitely they will be having carbonyl and hydroxyl. There. So hydroxyl is OH, as you can see over here, and carbonyl is this one. All right. So that's why option C is the correct answer. Now, the next question is question number 55. So question number 55 says that which of the following is not a product of light reaction of photosynthesis? All right. So which is not considered to be as the light reaction of photosynthesis? So let me tell you that photosynthesis basically has two types of reactions. One is the light reaction, the other is the dark reaction. The light reaction of photosynthesis, it basically involves some of the byproducts are released in case of the light reaction. So light reaction basically involves photolysis of water, that is splitting of water molecules that releases oxygen. All right. Then there is synthesis of ATP, which is also being involved. Then there is synthesis of NADPH also, which is being involved. So when you just look into the products of light reaction, so products of light reaction are ATP, NADPH, and oxygen. All right. So that is the case. So we all know that through the process of phosphorylation, all right. So cyclic or non-cyclic photophosphorylation, this ATP is synthesized. So you can say that this is synthesized via, so synthesized via cyclic and non-cyclic photophosphorylation process of light reaction, So I'm telling about ATP. So ATP is usually synthesized via cyclic and non-cyclic photophosphorylation of light reaction. While this NADPH, if you just consider about NADPH, so NADPH is synthesized via non-cyclic photophosphorylation. So synthesized via non-cyclic photophosphorylation. Non-cyclic photophosphorylation. All right. However, the cyclic photophosphorylation does not produce NADPH. So it is synthesized only via non-cyclic photophosphorylation, which is also considered to be as Z scheme. This is also known as Z scheme. All right. Now, oxygen, we all know that oxygen is produced via photolysis of water. So this is produced via photolysis of water. That means splitting of water. All right. So these are the products which are released or which are the products of light reaction. So let us take a look. ATP synthesized, NAPH. There is no other products, none of the products in the name of NAPH which is released. It might be NADH, but it might not be NAPH. So definitely this is the correct answer with regard to the question, which is not a product of light reaction. Oxygen is also a byproduct. NADPH is also produced and ATP is also produced. We have all seen over here this entire thing. All right. So that's why the correct answer is option B. So the next question is question number 56. So this question says that the stomatal movement is not affected by temperature, light, CO2 concentration, and oxygen concentration. Now, this question has been asked from the chapter of transport and plans from your standard 11. And let me tell you that stomata are basically those structures that helps in the process that are actually found in the leaf epidermis. These are the openings found in the leaf epidermis and through which the process of transpiration and gaseous exchange takes place. All right. So various factors affects the stomatal movement or opening or closing actually. So the factors like temperature, all right, and temperature, light, and CO2 concentration. CO2 concentration. So these factors affects stomatal movement. So these factors are responsible for stomatal movement. Now let me tell you that in temperature, it is basically the high temperature. So high temperature induces stomatal opening. High temperature induces stomatal opening, while low temperature will induce its closing mechanism. All right. On the other hand, the presence of light will induce stomatal opening. So presence of light causes stomatal opening. And on the other hand, if you just look into the CO2 concentration, so low CO2 concentration is responsible for stomatal opening process. That means low CO2 induces stomatal opening. All right, so these are the reasons. So definitely these factors affect stomatal movement or stomatal opening and closing mechanism, but it is the oxygen concentration that does not affect the stomatal movement or stomatal opening and closing mechanism. So the correct answer is option D over here, because oxygen concentration has no relationship with stomatal opening or movement. Oxygen concentration does not affect stomatal movement. So this is not going to affect the stomatal movement. So that's why the correct answer is option D over here. All right. The next is question number 57. Now the question number 57 says that the Golgi complex participates in. All right. So the Golgi complex participates in fatty acid breakdown, formation of secretory vesicles, activation of amino acids, and respiration in the bacteria. Now, let me tell you that this question has been asked from the chapter of cell, the unit of life from your 11th standard. And if you just consider the functions of Golgi complex, so Golgi complex are one of those membrane bound cell organelle which is actually responsible for forming secretory vesicles. All right, because they releases secretory vesicles from, they have basically two phases. One is the cis phase, other is the trans phase. So those secretory vesicles, they are released from the trans phase of the Golgi vesicles. So Golgi complex is involved in formation of secretory vesicles. And these secretory vesicles, they basically contains products from the, coming from the endoplasmic reticulum. All right. So, and these vesicles contains products from endoplasmic reticulum, because these Golgi complex, they are in association with ER. That's why. So these vesicles, they will be containing products from the ER and are then budded out and are then released or bud off from the trans phase of Golgi. So they are basically bud out from the trans phase of Golgi after maturation. And they are released to their target site. All right. So we can say that the option B is the correct answer over here. The first option which says fatty acid breakdown, so this is basically a function of peroxisome. Activation of amino acids, no, this is not the function. And respiration in bacteria, this is carried out by mesosome. All right, so definitely these are not the options. These all are not the options. So the correct answer is option B over here. Now, question number 58. Question number 58 says that which of the following is true for nucleolus? All right. Larger nucleoli are present in the dividing cells. It is a membrane-bound structure. It is a site for active ribosomal RNA synthesis. It takes part in the spindle formation. Now, let me tell you that this question has been asked from the chapter again, from the chapter of cell, the unit of life, from your standard 11. And nucleolus is one such structure which is found inside nucleus. All right. So, and since this is found inside nucleus, so it is not a membrane-bound structure, rather it is a non-membranous structure. So, nucleolus is found inside the nucleus. This is basically found inside the nucleus of a cell, and this nucleolus, this is basically a membrane. It is actually a non-membrane, not membrane. This nucleus is a membrane-bound organelle, but nucleolus is a non-membranous structure. So that's why this is a non-membranous structure which is actively involved in the synthesis of ribosomal RNA, which is actively involved in synthesis of rRNA, which is called as ribosomal RNA. All right. So that's why if you just look into the options that has been given, the correct answer is option C over here. membrane bound structure won't be correct. Then it is a larger nucleoli are present in the dividing cell. No, this is not such an option like this. And it takes part in spindle formation. Again, no. All right. So that's why it is basically found inside the cell, that is involved in the actively involved in the synthesis of RR. All right. Question number 15. Question number 59. So this question says that stomata in the grass leaf, they are dumbbell shaped, kidney shaped, barrel shaped, or rectangular shaped. So this question has been asked from the chapter of transportation in plants of your standard 11. And again, this is a very direct kind of a question that has been asked, because let me tell you that grass is basically a monocot plant. So grass is a monocot plant. All right. And in case of monocotyledonous plants, the stomata found is of dumbbell shape. And in case of monocotyledonous plants, the stomata is dumbbell shaped. All right. So if you just take a look into the structure, so this something looks like this. It is usually dumbbell shaped kind of a structure which is present. Like this. All right. And again, they have the presence of subsidiary cells and epidermal cells around them, like this. All right. And the microfibrils, cellulose microfibrils, they are present over here. And this some things look like this, this category. All right. So this is dumbbell shaped stomata, or dumbbell shaped rather guard cells, we can say, because this structure basically represents dumbbell shaped guard cells of stomata. While in case of dicot, the stomata structure is kidney shaped. All right. So it is a very direct question, as I told you. So that's why the correct answer is option A over here, which is dumbbell shape. Kidney shape is found in case of dicot plants. This is in case of dicot plants. All right. Now, the next is question number 60. So question number 60 says that the stage during which the separation of the paired homologous chromosome begins is, all right. So the paired homologous chromosome is now going to separate. All right. So pectine, diplotin, zygotin, and diakinesis. So this question again has been asked from the chapter of cell cycle and cell division of your standard 11. All right. And this is a question that has come up from one of the phases of the prophase 1 of meiosis 1. All right, these are basically the stages of the prophase 1 of meiosis 1. So let me tell you that the process of separation of paired homologous chromosomes, if you just look into the sequence, let me tell you first the sequence. So the sequence of phases of meiosis, of prophase 1 of meiosis 1. Prophase 1 of meiosis 1. So the first is leptotene. then zygotine, then pecitine, then diplotene, and the last is dikinesis. So these are the sequential stages. All right. So in case of the process where it starts with leptotene and it ends up to dikinesis, all right, out of all the stages, so the leptotene is basically characterized by chromosomal condensation. So this is the process where chromosomal condensation occurs. All right. Now, zygotane is characterized by synapsis and synaptonymal complex. So, formation, or rather, you can say synapsis and synaptonymal complex formation. So, synapsis is basically the process of formation of bivalents. So, synapsis and synaptonymal complex formation occurs over here. All right. And pecitine is characterized by, again, crossing over between bivalents or between the paired homologous chromosomes. Bivalents. All right. Then diplotene is characterized by, once the crossing over has taken place, now there will be the separation of those bivalents or separation of the paired homologous chromosome. That means it is basically characterized by the process where the chiasma formation occurs. All right. So chiasma formation and separation of the homologous chromosomes. So you can say chiasmata formation. And once the chiasma is formed, there will be separation of paired homologous chromosome. This is going to actually start. Homologous chromosomes. And the last, which is dikinesis, this is marked by terminalization of chiasma. Terminalization of chiasma. All right, so this is the case. So as you can see on your screen, so diplotene is basically related with the separation of the paired homologous chromosome once the crossing over has taken place. So that means option B is the correct answer over here. All right. Now, question number 61. Question number 61 says that which of the following is commonly used as vector for introducing the DNA fragment in the human lymphocytes? Commonly used as a vector in introducing the DNA fragment in the human lymphocyte. So let me tell you that this question has been out from the chapter of biotechnology and its applications from standard 12. All right. And the commonly used vector in case of the human lymphocytes are basically the retroviruses, because this is the classic case of ADA deficiency that you people might have studied it as well. All right. So where the retrovirus, these are all the vectors, that cloning vectors that can be used for cloning. But in case of human lymphocyte, if you specifically talk about, then it is the retroviruses. All right. So we can say that retrovirus is commonly used as cloning vector for introducing DNA fragment in human lymphocyte. And as I told you that this is a classic case of ADA deficiency, that means gene therapy concept, human lymphocytes. All right, so how the process takes place? So it's a classic case of gene therapy. So in this case of gene therapy, the lymphocytes, the particular patient who is suffering from ADA, that means adenodeaminase deficiency enzyme, which is responsible, the enzyme which is actually responsible for immunity, the patient is suffered from that enzyme, that the patient lacks that enzyme. So what is done is that the lymphocytes from the blood vessels, from the blood of the patient, that is taken up. All right, so we can just write that lymphocytes from the blood of the patient is taken and grown in culture outside the body. So it is exactly grown in culture outside the body. Then a functional gene, which is ADA-cDNA. Then a functional gene that is complementary to that ADA actually. So that's called as ADA-cDNA. The gene is called ADA-cDNA, is introduced into the grown cultures. So is introduced using a retroviral vector. Retrovirus has vector, and that is subsequently returned to the patient. Into these lymphocytes which are subsequently returned to patient. So that's why the patient actually needs, since these lymphocytes, they have a particular lifespan. So that's why the patient having such kind of treatment or given such kind of treatment, they require periodic infusion of such genetically engineered lymphocytes to be introduced into their body. All right. So that's why here we are basically using retrovirus as a vector. All right. So that's why the option A is the correct answer. Now, question number 62. So question number 62 says that use of bioresources by multinational companies and organizations without authorization from the concerned country and its people is called bioinfringement, biopiracy, bioexploitation, biodegradation. Now again, this is a very direct question that has been asked from the chapter of your biotechnology and its applications. So this is a definitive question that has been asked. So let me tell you that the process by which the bioresources are illegally used without any authorization or permission from the concerned company or organization, and from the country as well. So that illegal use is considered to be as biopiracy. So we can say that option B is considered to be the correct answer, because biopiracy is the term. So this is the term used to refer the use of bioresources by multinational companies and organizations. All right. So by multinational companies and organizations without prior authorization from concerned country. And its people. So this is the case. All right, without any kind of compensatory payment, you can say. All right, so that's why the correct answer is biopiracy. Now, question number 63. So question number 63 says that in India, the organization responsible for assessing the safety of introducing genetically modified organism for public use is ICMR, which is Indian Council of Medical Research, then Council of Scientific and Industrial Research, which is CSIR, Genetic Engineering Appraisal Committee, then a Research Committee on Genetic Manipulation. All right. Now, again, this is a question that has been asked from Biotechnology and its Principles chapter from your Standard 12. And let me tell you that before any decisions or any validations which is being carried out on GM research, all right, so the Indian government has actually set up an organization, and that organization is considered to be as Genetic Engineering Approval Committee or Appraisal Committee. So in case of any GM researches, that means genetically modified researches which are being done in India, so for the safety, for the validity of the GM research and for the safety of introducing those GM organisms to the public services, this is very, very important that the GX should undertake all those validations. So that's why C is the correct answer. So we can like write it as Indian government Indian government has set up an organization called GAC, all right, which is known as Genetic Engineering Appraisal or Approval Committee, you can say. All right. Now, this organization will make decisions regarding any kind of validation. So, this makes any decisions regarding the validation of GM research and the safety of introducing the GM organism for public services. And the safety for introducing genetically modify organisms for public services. All right. So that's why. So that's why the answer is option C over here. Now, question number 64. So question number 64 says that the correct order of steps in PCR, that means polymerase chain reaction, is. So this question has been asked from the chapter of biotechnology principles and processes of your standard 12th. And let me tell you that PCR is one such mechanism which is known to amplify a particular fragment of DNA or particular segment of DNA. So PCR is a technique of amplification of DNA. DNA segment, you can say, basically. So this is basically a process where you increase the copy number of a DNA segment. That means amplification of DNA segments means to make multiple copies of a gene or DNA, you can say, or gene of interest, you can say, basically, of gene of interest or DNA. And this is performed in, since this is performed under laboratory conditions, so we call it as in vitro technique. All right. So PCR basically involves three important, very, very important steps. So the sequence basically includes sequence to perform PCR, sequence to perform PCR, that means polymerase chain reaction. So this basically requires, the first step is the denaturation, followed by annealing, followed by extension. So this is the three steps. So denaturation, annealing, and extension. All right. So denaturation is basically a process where the two strands of DNA are separated from one another. Annealing is a process in which all the substrates, or you can say all the requirements are added into the enzyme, into the mixture for carrying out the process. And then extension means a particular DNA segment will be extended. All right. So this is the steps. So let us take a look. So it first starts with denaturation, then annealing, and then extension. So as you can see, option C. So this is the correct answer. All right. The next is question number 65. So question number 65 says that you have to select the correct match regarding the following options that has been given. So the first is ribozyme, which is a nucleic acid. Then F2 into recessive parent, if you are just going to cross it, then this will result into a dihybrid cross. Then Gregor John Mendel induced the transformation experiment. Then there is Thomas and Morgan. He proposed the transduction experiment. Now let me tell you that you have to select the correct match. All right. So out of all the options that has been given, the correct match is the ribozyme. And ribozyme, let me tell you that ribozyme is basically a catalytic RNA. So this is a catalytic RNA. All right. And since this is a catalytic RNA, so that's why this will be considered to be as a nucleic acid. So which is a nucleic acid. So definitely this is going to be the correct option. All right. Now, when you are going to, like here, it has been written that F2 will be crossed with the recessive parent, when you're going to cross F2 with that of the recessive parent, so definitely this is a category of test cross, not the dihybrid cross. So definitely this is not the correct answer. All right, this is a wrong option. Then Gregor John Mendel, he is considered to be the father of genetics. So he don't formed, or he don't propose the process of transformation. All right. So transformation experiment is one such experiment which is actually proposed by Griffith. All right. And then Thomas Hunt Morgan is basically related with the chromosomal theory of inheritance. All right. So we can say that when F2 is crossed with recessive parent, this is basically a correct match. This is the correct match. Correct match. Incorrect matches, if you are going to see it, then F2 with recessive parent, this is called as test cross, not the dihybrid cross. All right. Then Gregor John Mendel. G. Mendel is basically related with genetics. So it has nothing to do with transformation. So transformation was proposed by Griffith. Transformation proposed by Griffith. So you might have heard it about the Griffith experiment as well. And the last is T.H. Morgan. So Thomas Hunt Morgan is basically related with chromosomal theory of inheritance. So he basically proposed chromosomal theory of inheritance. All right, so definitely this is also not the correct answer. The correct answer is option A over here. All right, now question number 66. So question number 66 says that a new variety of rice was patented by a foreign company. Through such varieties has been present in India for a longer time, and this is related to. All right, so this is talking about a new variety of rice. Now, this question has been asked from the chapter of strategies in enhancement, strategies of enhancement in food production from your standard 12. So this is again a very, this is again a very important and a direct question that has been asked actually. All right, so we can say that, in the year of 1997, the patent rights on basmati, this one, say, since basmati is an Indian origin rice species. However, an American company, without any kind of consent, or without any kind of authorization from the India, they got their patent rights on basmati rice actually, and through US Patent and Trademark Office. So that was actually has been variety of India. So we can say that basmati is considered to be that new variety of rice that was patented by a foreign company, and that is an American company. All right. So we can just write that an American company in 1997 got patent rights on basmati. Basmati, we all know, is a rice variety, and India has produced approximately more than 200 varieties of basmati rice. That's why basmati, through US Patent and Trademark Office. All right. So that has actually been derived from India's farmers variety. So that has actually been derived from India's farmers varieties, because India is considered to be as a larger exporter of rice. India is a country where it exports large amount of rice or large varieties of rice actually. That's why. And it is richest in the world, and 27 documented varieties of basmati are grown in India actually. So you can say that 27 documented varieties of rice are grown in India. All right. So Indian basmati was also, crossed with the semi-dwarf varieties as well, and was that's why considered to be as a novelty, or you can say invention, in the genetically modified crop of Indian variety. All right, so that is the case. While on the other hand, if you just see into if you just look into the other options, which is CO667, Sarbati, Shonora, and Larmarojo, these are actually the varieties of wheat. So they have nothing to do with rice. These are wheat varieties. So definitely these cannot be the options. All right, so the only option left is basmati. So answer option C is the correct answer. Now, the next is question number 67. Now, question number 67 says that you have to select the correct match. Alec Jeffrey, Streptococcus pneumoniae, Alfred Hersey, and Martha Chase, they proposed TMV. Then Francis Jacob and Monod, they proposed Lacoperon. Then Matthew Michelson and Franklin Stahl, so they basically proposed the Pisum sativa. Now, let me tell you that the correct match out of all the matches is basically this one. That means option C over here. All right. So the correct match is Francois Jacob and Monod. And Monod, who proposed the Lacoperon. So they were the inventor of Lacoperon method. All right, so this is the correct match. While the incorrect matches, they are basically, the incorrect matches are so this basically includes the very first important option, which is Alec Jeffreys. So Alec Jeffreys was the person who proposed the DNA fingerprinting technique. All right, so he proposed Alec Jeffreys. So he proposed DNA fingerprinting technique. All right, then B, which is considered to be as Matthew Michelson and Franklin Stahl. So they proposed, they do not discover the TMV, but they actually proposed the semi-conservative method of DNA replication. So Hersey and Chase, Hersey and Chase, they are responsible for the process, or they actually proved to be they were basically responsible for proving DNA as the genetic material through their experiment. All right, of labeled nitrogens and phosphorus. So they proved DNA as genetic material. Genetic material and not protein. And then comes up the last one, which is Messelson and Stahl, which is D1, which is Messelson and Franklin Stahl. They are responsible for proposing the semi-conservative method of DNA replication. So semi-conservative method of DNA replication in E. coli. All right, so this is the case. So that means the correct answer is, or the correct match is option C over here. Now, the next is question number 68. Question number 68 says that which of the following has proved helpful in preserving pollen as the fossils? All right. So is it pollen kit, cellulosic intine, sporopollenin, and the oil content? So this question has been asked from the chapter of your sexual reproduction in flowering plants from your standard 12th. Now let me tell you that in preservation of fossils, pollen grains usually have two layers. All right. So pollen grains, if you see the structure of pollen grains, so they usually have two layers surrounding them like this. And it is a two-cell stage if you like draw it. So this layer is considered, the outer layer is called as exine, and this is usually made of sporopollenin, which is a chemical substance, one of the hardest chemical substance known, and no enzyme or acids or alkalis or any chemical can degrade the sporopollenin. So it is sporopollenin, while this internal layer is considered to be as intine, which is usually made of cellulose and pectin. So this is made of cellulose and pectin, and this is a pollen grain. All right. So definitely the cellulosic intine won't be the option, the pollen kit won't be the option, and the oil content again won't be the option, because this pollen kit is basically responsible for, let me tell you first regarding the sporopollenin, and then we'll go ahead. So we can say that sporopollenin is one of the hardest chemical substance known. Chemical substance known. Then no acids, as I told you, that no acids or alkalis or enzymes or temperature treatment can degrade sporopollenin. And because of this nature, that this cannot be degraded by any acids, alkalis, or enzymes or temperature, that's why they are preserved as fossils. That's why it is due to sporopollenin that pollens are preserved as fossils. Remember. All right. Now, regarding the other options that has been given, pollen kit is basically a substance that surrounds the sporopollenin, but it has nothing to do with the preservation part, because it is actually helpful in attracting the insect. So this basically helps in insect pollination. It is going to attract the insects, so that's why it's helped in insect pollination. Then oil content has nothing to do with the preservation of pollen, so they have no role in pollen preservation, you can say. Pollen preservation. And the last one is the cellulosic intine. As I told you, that cellulosic intine is basically the inner layer of the pollen grain, which is known as intine, and it is made of cellulose and pectin. So this has also nothing to do with the preservation of fossils, pollens as fossils. So that's why the correct answer over here is the option 3, C, which is sporopollen. Now, the next is question number 69. So the question number 69 says that the experimental proof for the semi-conservative replication of DNA was first shown in a fungus, a bacterium, virus, and plant. Now, this question has been asked from the chapter of molecular basis of inheritance from your standard 12. And let me tell you that this semi-conservative method of DNA replication, which was proposed by Franklin and Stahl, Messelson and Stahl, Franklin, Messelson, and Stahl. So that was actually proposed for the very first time. It was proposed in case of a bacterium, and that is E. coli. So that is Messelson and Stahl, they proposed it. And this semi-conservative replication of DNA, it basically states that in the process of DNA replication, there is one DNA strand which is conserved while the other is newly synthesized. So we can say that semi-conservative replication of DNA was proposed by, so this was proposed by Messelson and Stahl, Matthew Messelson and Franklin Stahl. All right, which stated that out of 2 DNA strands, one strand is conserved, one strand is conserved. Conserved strand basically means that it is a parental strand. So one strand is conserved, that is, it is a parental strand. It is a parental strand. And the other is a newly synthesized strand. And the other is a newly synthesized strand. So that's why the term semi-conservative, it has been written over here. All right, semi-conservative means half conservative, in which the one strand will be conserved, that will belong to the parental strand, and the other strand is a completely newly synthesized strand, which is synthesized in complementary to the parental strand. So this is called semi-conservative method of DNA replication. And this for the very first time was shown in, so it was shown in a bacterium that is E. coli. So this is the case. All right. So that means option B is the correct answer. Now, question number 70. The question number 70 says that which of the following pairs is wrongly matched? So you have to find out the wrongly matched pairs. So it says that the starch synthesis in P. All right. So multiple alleles. So does it indicates multiple alleles? Then ABO blood grouping is an example of codominance. Then TH Morgan is with linkage, and exotype of sex determination is found in case of the grasshopper. All right. So out of all the options that has been given over here, the wrongly matched pair is basically the wrong match is the first option, which is starch synthesis in P. So this is not a case of multiple allelism, but it is a case of pleiotropism. All right. So the wrong match is starch synthesis in P, which is a case of pleiotropism. That means a single gene controlling several characters or trait. So that's why. All right. So this is a case of pleiotropism. We can write like this. All right. On the other hand, the ABO blood grouping, which is a case of codominance, this is completely correct. This is a correct option. Yes, they indicate this is a correct match because they indicate a process of codominance where there is no dominance or recessive relationship. That's why. Then TH Morgan is related with linkage and recombination, you can say. So this is also correct. So the concept which is based on chromosomal theory of inheritance. Then exotype of sex determination is found in case of insects, particularly the grasshopper case. You might have studied it also as well. So this is also correct. All right. So the incorrect match is option A. All right. Now, the next question is question number 71, which says that offsets are produced by. All right, meiotic division, then mitotic divisions, then parthenogenesis and parthenocarpy. So this question has been asked from the chapter of your sexual reproduction in plants, and this is basically the first chapter of your 12th standard. All right. So let me tell you that offsets are basically those categories, or offsets are considered to be as the vegetative part of the plant bodies. All right. So these offsets, they basically refer or they represent as the vegetative structures or propagules of the plant. So offsets are vegetative part or propagules of plants. All right. So these offsets, since these are the vegetative part or propagules of the plant, so they generally represent that they will be undergoing any vegetative part of the plant, which will be undergoing the process of reproduction. So that will undergo the process of asexual reproduction. So this basically indicates. So this indicates that offsets are used in asexual mode of reproduction. Asexual mode of reproduction. Now, if there is asexual mode of reproduction, so we know this fact very well that in case of the asexual method of reproduction, the process of division that is involved is only the mitotic division. All right. So here we can say that in this process of asexual reproduction, so technically we can say that offsets are formed by mitotic divisions. These are actually formed by mitotic division, because meiotic divisions, they are a part of sexual reproduction, and offsets are vegetative propagules. So definitely they won't be, this process of division won't be involved in case of offset production. So this option is the wrong answer. So mitotic divisions has been given. So this is actually the correct answer. And let me tell you regarding the parthenogenesis and parthenocarpy. So parthenogenesis is basically the process. This is also a mode of asexual reproduction, and this is usually found in case of certain categories of insects and animals, where only the female organism undergoes development and produces, offsprings without fertilization, we can say. So we can say that parthenogenesis is a process of formation of embryo from ovum or egg. That means only the female organism is involved, not the male one, without fertilization. This is without fertilization. All right. And regarding the parthenocarpy, so parthenocarpy is a process of producing seedless fruits, because this is a process in which the fruits are formed without the fertilization. So fruits formed without fertilization of ovum. All right, so this is the case. So definitely these two also are not the option. So the correct answer is option B over here. Now, question number 72. Question number 72 says that you have to select the correct statement. Franklin Stahl coined the term linkage. Punnett square was developed by British scientists. Transduction was discovered by S. Altman, and spliceosomes takes part in translation. So this question, these statements, they have been asked from the chapter of your, it is a mixed kind of, questions that has been asked from different chapters actually. So that's why we can say that if you just find out the correct statement, then the correct statement is basically the option B over here, which says that Punnett square was developed by a British scientist. All right, and that is C. Punnett. So this is the correct statement. So we can say that the correct statement is option B, which says that Punnett square was developed by a British scientist called Reginald C. Punnett. So this is a, that's why this is a true statement actually. Punnett. All right. While the other statements, those are considered to be as the incorrect statements. The incorrect statement of, because the very first important statement that says that Franklin Stahl coined the term linkage. So this is the wrong statement, because Franklin Stahl was the scientist who proved the semi-conservative method of DNA replication. So Franklin Stahl proved semi-conservative mode of DNA replication and not the linkage. So this is also wrong. Then there is transduction was discovered by S. Altman. So let me tell you that transduction is one of the methods of sexual reproduction in bacteria or in prokaryotes. So this transduction was discovered by, so transduction was discovered by Zinder and Lederberg. So definitely this is also the wrong one. And the last one, which says that spliceosomes takes part in translation. So spliceosome doesn't take part in translation because these are an important component that takes part in the post-transcriptional gene modification or post-transcriptional modificational changes occurring in the eukaryotes. So we can say that spliceosome formation is a part of post-transcriptional change in eukaryotes. So this has nothing to do with the translation. All right. So that's why option B is the correct answer. Now, question number 73. Question number 73 says that which of the following flowers only once in its lifetime? So is it bamboo species, then jackfruit, then papaya, then mango? So this question has been asked from the chapter of your sexual reproduction in flowering plants from your standard 12. And let me tell you that the flowers that only, the plants that flowers only once in its lifetime and then they die, those flowers are, or those flowers are considered to be as the monocarpic flowers. All right. So we can just consider that plants that flowers only once in lifetime and then dies. So this is called monocarpic flowers. These are called monocarpic flowers. And the best example of monocarpic flower is bamboo. Is the bamboo tree, because bamboo tree flowers generally once in its lifetime, and that too lifespan ranging from 50 to 100 years. So that is the case. So bamboo generally flowers once in its lifetime. Generally flowers once in its lifetime after 50 to 100 years. All right. So that's why option A is the correct answer. While jackfruit, papaya, and mango, these are the categories of the flowers or the plants, you can say, where the flowering occurs many times in their lifetime. All right. So they flower. Most of them, like many of them, are biennials. They can be perennial or annuals. All those things. Now, So the next is question number 74. Question number 74 says that niche is, all right. So all the biological factors in the organismic environment, the physical space where an organism lives, the functional role played by the organism where it lives, and the range of temperature that the organism needs to live. Now, this question has been asked from the chapter of your organism and population from your standard 12 ecology section. All right, now let me tell you the definition of niche. Niche is basically considered to be as a physical space that an organism occupies. All right, that means the physical habitat, you can say. So a physical space occupied by an organism, and not only the physical space, but what will be the functional aspect or functional role of that particular organism in the community, that is also included in the niche. So the physical space occupied by an organism along with its functional role in the community. So along with its functional role in community. So this is considered to be as ecological niche, and this term was given by Grinnell. This niche term was given by, so term given by J. Grinnell. All right, so this is the case. So let us take a look into the statements again. So all the biological factors in the organismic environment, again, no. The physical space where an organism lives, so this is considered to be as habitat. All right, so definitely this won't be an option. Then the functional role played by the organism where it lives. Yes, this is the correct one. The range of temperature that the organism needs to live, no. Again, so that's why the correct answer is option C over here. Now, question number 75. Question number 75 says that in stratosphere, which of the following elements acts as a catalyst in degradation of ozone and release of molecular oxygen? Now, let me tell you that this question has been asked from the chapter of your environmental issues of standard 12. All right, and this is asking about the ozone formation and degradation one. So basically, the one thing which is very, very important in forming or degrading ozone are the UV rays, because UV rays are going to these ozone, that means O3 molecule, or you can rather say the degradation of ozone. These UV rays, they act on the chlorofluorocarbons. All right, so if you just note down the explanation, then UV rays acts on CFCs. That means they will be acting on the chlorofluorocarbon. Now, once they will be acting on the chlorofluorocarbon, they are going to release the chlorine atoms, after which they release chlorine atoms. All right, and this chlorine is going to react with the ozone that is present in the stratospheric environment. So this chlorine atom reacts with ozone and thus splitting ozone. So thus it is going to split ozone into oxygen and nascent oxygen. So this basically splits ozone into two parts, that is oxygen and nascent oxygen. All right, so we can say that out of all the options that you can see over here, so it's carbon, chlorine, oxygen and iron. Iron has nothing to do, oxygen also, oxygen is released as a byproduct. So definitely this won't be acting, and then carbon also won't be acting. So it is the chlorine which is responsible for degradation of ozone, and ultimately it basically breaks up. The reaction goes like this. The reaction will be, if this is O3, so here the chlorine atoms will be acting on it, and it will split ozone into two categories. This one. So this is basically ozone, and this is nascent oxygen. All right. So that's why it's the chlorine. That means option B is the correct answer. Now, question number 76. Question number 76 says that what type of ecological pyramid would be obtained with the following data. So secondary consumer is 120 gram, primary consumer is 60 gram, primary producer is 10 gram. Inverted pyramid of biomass, pyramid of energy, upright pyramid of biomass, upright pyramid of number. Now let me tell you, this question has been asked from the chapter of your ecosystem of standard 12. All right. Now from the given data, if you see, so the secondary consumer is 120 gram. So this is 120 gram. Okay. Then primary consumer is having 60 gram. Is having 60 gram. And primary producer is having 10 gram. So that means at the producer level, if you will see to it, then it is having the less quantity of food. Less quantity of food. So this is depicting what? This from the given data, it is depicting, since this is less. See, primary producer, it is less. All right. So that means they are synthesizing. Primary producers are the producers which will be synthesizing food, and they are synthesizing food in a very less amount. All right. So but in general, if we say or if we look into the pyramid, it is usually upright. So this pyramid cannot be upright. From this data we can say that this pyramid cannot be upright. So this pyramid can't be upright. So this pyramid actually can't be upright, because from the data, if it would had been upright, then this would have been greater. That means the primary producer would have been greater. All right. And then the primary consumer, and then the secondary consumer would have been the least. But here the entire data is opposite. So definitely the pyramid of energy cannot be the option, because pyramid of energy is always upright. All right. So pyramid of energy is always upright. So definitely this is not the option. Then there is inverted pyramid of biomass. See, upright pyramid, as I told you, that this pyramid can't be upright from the given data. So there are two upright options has been given. So definitely this can't be the option. All right. And yes, because this is the lesser one as compared to the given data. So inverted pyramid of biomass can be the correct answer, because if you just draw it, inverted pyramid of biomass, so this is going to be like this. That means producer is the least one. Then here, so here will be primary producer, here will be your primary consumer, and here will be secondary consumer. So here it's 160, I guess. It's 120, 60, and 10. So this is 160. This is 120. Sorry, it's 120 and 160. Wait. So it's 120 here and 60 here, and of course 10 here. All right. So this is the inverted pyramid of biomass that we can see. An inverted pyramid of biomass is obtained in case of aquatic ecosystem. That means pond ecosystem. So this is inverted pyramid of biomass, or you can say depicts inverted pyramid of biomass, which is found in pond or aquatic ecosystem. Where the phytoplanktons are the least in number, then primary consumer is being represented, like this will be represented by phytoplanktons, because this is according to biomass actually, that's why. Then this will be zooplanktons, and the secondary consumer is represented by small fishes. All right, so this is the case. So that's why the option A is the correct answer. Now, question number 77. Question number 77 says that which of the following is a secondary pollutant? All right, so this question has been asked again from the chapter of your environmental issues. All right, now let me tell you that there are basically two categories of pollutant. Primarily, there are two types of pollutant. One is considered to be as the primary pollutant, which is released directly into the source, and the other is called as the secondary pollutant, which is not released directly into the source, but they react with certain gases or chemicals that are already found in the atmosphere, and then they are released in the nature. All right, so this is the case. So out of all the categories that has been given, all right, so primary pollutant, in case of primary pollutant, if you are going to see, so this basically involves CO2, SO2, and carbon monoxide, which is CO. These are all the primary pollutants because they are released directly into the source. However, CO is released, this CO is carbon monoxide, so this is primarily released from the CO2. If there is incomplete combustion of fossil fuels, then that releases carbon monoxide. So all these are primary pollutant, while the secondary pollutant is ozone. The only left option is ozone, because these secondary pollutants, these ozone, they are of two categories. They can either be good ozone, they can either be bad ozone. So the bad ozone is found in the tropospheric zone, and once this bad ozone is splitted by chlorine atoms, as I told you, they will be released into the oxygen and nascent oxygen, and so they are harmful. So that's why the secondary pollutant is ozone. So this, as I told you, that secondary pollutant are formed as a result of reactions from the primary pollutant. So these are formed as a result of reactions with primary pollutant. While primary pollutant, they are directly released into the source. So that's why option C is the correct answer. Now, next is question number 78. Question number 78 says that World Ozone Day is celebrated on. All right. So World Ozone Day is basically celebrated. This question again is a very direct question that has been asked, and it is from the chapter of your environmental issues. So World Ozone Day is basically celebrated on 16th September. All right. So World Ozone Day is celebrated on 16th September. So this is basically celebrated on 16th September, while 5th June is considered to be as the World Environment Day. This is known as World Environment Day. Then 21st April is considered to be as the National Yellow Bat Day. So this is National Yellow Bat Day, and 22nd April is considered to be as National Earth Day. All right, so that's why the correct answer is 16th September. That means option D. Now, question 79. Question 79 indicates that natality refers to death rate, birth rate, number of individuals entering a habitat, number of individuals leaving the habitat. So basically, this question has been asked from the chapter of your organism and population of standard 12. And again, this is a very direct question and a definitive question which has been asked. So let me tell you that natality is known as birth rate. So natality is known as birth rate. All right. That means number of births taking place in a population so number of births taking place in a population in a given time period, you can say. All right, so this is called as birth rate. So that's why the correct answer is option B. Now, death rate is known as mortality. So this is known as mortality, which is the death rate. All right. While number of individuals entering a habitat, so this is known as entering a habitat, basically. So this is known as immigration. This phenomena is known as immigration, while the number of individuals leaving the habitat is known as emigration. Emigration. So immigration always increases the population, increases the number of population, while emigration always reduces the number of population. All right. So that's why the correct answer is option B over here. Now, question number 80. Question number 80 says that you have to match the items in the given column 1 with those in the column 2 and select the correct option given below. All right, so let us take a look into the columns. So the first option is herbarium. So herbarium is basically a collection of, plant specimens. And those plant specimens, it's a storehouse of basically plant specimens, you can say, where those plant specimens, they are dried, pressed, and preserved on sheets. So we can say that herbarium, it is a storehouse of collected plant specimens where they are dried, pressed, and preserved on sheets called herbarium. All right. So it is having a collection of preserved plants and animals. No, here only plants are preserved. You should remember this, that only plant specimens are preserved. So that's why a list that enumerates methodologically all the species found in an area with brief description aiding the identification. Then there is a place where dried and preserved plant specimens mounted on sheet are kept. So this is the correct answer. All right, so this is going to match up with option A, you can say. This is with option A. All right. Then there is key. Key is basically, you can say, a taxonomical aid, that helps in basically identification of various kinds of taxonomic group on the basis of similarities and differences. So for key, you can say that it's a
taxonomical aid. So this is basically a taxonomical aid used for identification of various taxa on basis of similarities and differences. All right, so this is about key. So if you just look into it, then you can say that if you look into the options, so it's a booklet containing a list of characters and their alternates which are helpful in identification of various taxa. So yes, this is the correct answer. So that's why this is going to match up with B. Okay. And then the option C is museum. Museum again, museum is something where both plants and animals are preserved. So it's a place of preserving plants and animals. So again, museum is the third option. So it's a place for preserving both plants and animals. All right, so this is going to match up with A, and the last option, this is going to match up with C, not A. This is going to match up with C, and the last option left is catalog. So catalog is basically a list that enumerates methodically that all the species found in an area with the description uh, along with the identification parameters that will help in its identification. So this is going to match up with D option. All right, so that means it basically involves alphabetic listing of the different species. That means from A to Z. So that is the case. So we can say that catalog is alphabetic listing of species that can either be of plants or animals. All right, so that is the case. So let us take a look into the options that has been matched up. So A is going to match up with third one. A is going to match up with third. B is going to match up with fourth. B is going to match up with fourth. Then C is going to match up with one, this one, and D is going to match up with second. So that means option third is the correct answer, this one. That means option D is the correct answer. All right. So the next question is question number 81, which says that you have to find out that which one is wrongly matched. Uniflagellate gametes, Polysiphonia, biflagellate zoospores, brown algae, unicellular organism, chlorella, and gamma cups are found in Mercantia. So this question has been asked from the chapter of your plant kingdom of standard 11th. And if you just look into the different options that has been given, so out of all the option, the wrongly matched or the wrong matching option is basically the first option, which says that uniflagellate gametes are found in Polysiphonia. And the reason for this is that Polysiphonia is actually a red algae. So this is actually a category of red algae. This is a red algae. And in case of red algae, all the sexual spores that are produced or the gametes that are produced, those are all non-flagellated, or the flagella is actually absent. But here it is saying that it is having a single flagella. Uniflagellate means that it is having a single flagellated gametes. So definitely this is a wrong option. So we can say that it's a red algae where the asexual spores asexual spores and gametes are, so they are basically non-flagellated. All right, so they are non-flagellated. That means the flagella is absent. So flagella is absent. So definitely this is a wrong match. All right, so this is the correct answer with regard to the question. Now, biflagellate zoospores are found in case of brown algae. So this is also correct, because in case of brown algae, the zoospores, they usually look like this. So they are usually having a flagella like this that can either be lateral or apical. So this is something like this, goes like this. And then unicellular organism is chlorella. So this is also correct and true, because chlorella is a green alga, which is a unicellular category. And gamma cups are found in mercantia. Again, this is also true, because these are asexual reproductive structures. So that is also considered to be as the correct one. All right. So that's why the correct answer for this question is option A over here. Now, question number 82. Question number 82 says that after karyogamy followed by meiosis, spores are produced exogenously in. All right. So it is asking about that after the process of karyogamy followed by the meiosis, that means followed by the reduction division, the spores will be produced exogenously in. All right. Now, let me tell you that basically, if you just, there are two types of categories in which the spores are produced. Spores can either be produced exogenously or they can either be produced endogenously. So if the spores will be produced exogenously, so this basically means that the spores will be produced outside the reproductive structures. So exogenously means spores production outside reproductive structure. So this is exogenously stands for. All right. Now, if you look into the different options that has been given, then the first option, which says neurospora. So neurospora is category of Ascomycetes, and in case of neurospora, they produces ascospores, and all those ascospores, they are they are the product of meiosis, but they are actually produced inside the ascus. So it's a member of Ascomycetes, where ascospores Ascospores are basically the sexual spores which are found in case of neurospora. So these are sexual spores. So ascospores are formed endogenously in ascus. So they are actually produced endogenously. Now, if you just look into the structure, so the structure something looks like this. So the spores are produced in sac-like structure, which is called ascus, and these are the spores. So this is basically ascus, and these are ascospores. So this indicates that the spores are produced inside the reproductive structure. All right. Then the second category is that of alternaria. So definitely this is not the option. The next category is alternaria. Alternaria. Now, alternaria is a member of Deuteromycetes. So this is a member of Deuteromycetes. And since this is a member of Deuteromycetes, so Deuteromycetes is one such class of fungi which do not produces any kind of asexual spores. So there is no chance of spore production either endogenously or exogenously. All right, so that will be the case. So we can say that where no asexual or sorry, no sexual spores are reported. No sexual spores are produced. Now, the third category is that of Saccharomyces. Again, this is also not the option. Saccharomyces is again, this is a unicellular category of Ascomycetian member. So Saccharomyces, which is also known as yeast. So this is a unicellular Ascomycetes. And since we all know that this is an Ascomycetian member, so definitely the spores will be produced inside the sac only. So where ascospores are produced endogenously inside ascus. Genously in ascus. All right. So that is the case. And then the last is, again, the only option left is agaricus. So agaricus, yes, this is the correct answer because agaricus is a member of Basidiomycetes. Agaricus bisporus. This is basically an edible fungi belonging to the member of Basidiomycetes. So it's a member of Basidiomycetes where basidiospores. So where basidiospores, and basidiospores are basically the sexual spores. So where basidiospores are formed exogenously. So these are produced exogenously on basidium. So this some things look like this. So we can have a picture also. So there is a structure which is formed like this. And they are always produced in sac-like structure like this. So this is the structure and the basidiospores, crown-like structure present, and the basidiospores, they are present like this. So these are basidiospores. Which is produced exogenously. This is the basidium. So each basidium bear 4 basidiospores. All right, so that's why the correct answer is option D over here. Now, question number 83. Question number 83 says that the winged pollen grains are present in. So mustard, cycas, pinus, and mango. All right. So this question has been asked from the chapter of your plant kingdom, and that is of your standard 11. And let me tell you that winged pollen grain is a characteristic feature, and anything which is a characteristic feature will only be found in a particular category of species. All right. So that's why the correct answer is pinus over here. All right. So winged pollen grains. So winged pollen grains, they are found in pinus. Are found in, and we know that pinus is a gymnospermic species, which is a gymnosperm. All right, so that is the case. And the structure something look like this. So it is having a circular structure like this, which is a pollen grain like structure, and it is usually having a wing like this. And you will see to it. So this is usually having a wing like this. So pollen grain with wings looks like this. So this is basically the extension part of the outer exine on the two lateral side to form the wing of pollen. And this is a characteristic feature of Pinus only. So this is winged pollen. Winged pollen. All right. While in case of mustard, cycas, and mango, mustard and cycas, these are angiospermic species, so definitely they won't be having the this one winged pollen grains. And as I told you that since winged pollen grain it is asking about, so it is a characteristic feature, and anything which is a characteristic feature will only be found in a single category of species. So that's why the correct answer is option C, and all other options are wrong over here. All right, now the next is question number 84. Question number 84 says that pneumatophores occur in halophytes, then free floating hydrophytes, then submerged hydrophytes, and carnivorous plant. Now, this question has been asked from the chapter of your morphology of flowering plants of standard 11. And let me tell you that pneumatophores are one such structures which are usually found in rhizophora plant species, you can say. And these plant species are basically the plant species that belongs to the helophytic category. All right, so indirectly we can say that helophytes like the helophytes are basically those categories of plant species that grows in extremely saline environment. So helophytes like mangroves that grows in extremely saline environment. So that grows in extremely saline environment have pneumatophores. So they have pneumatophores. And pneumatophores are basically those structures which provides or which helps in the respiration of the plant roots. And in such categories of pneumatophores, the thing that happens is that these roots they rises above the soil or that particular area where they are growing, and then these pneumatophores gets exposed, and that's how they help in the respiration process. So pneumatophores, you can say that these are the respiring roots. Respiring roots that helps in gaseous exchange. That comes above the soil and helps in gaseous exchange. All right. So this is the case. This is regarding the pneumatophores. All right, so that's why the correct answer is here, halophytes. Now, hydrophytes, definitely hydrophytes won't be having it. So this is again not the case because they are either it is free floating or submerged hydrophyte, and then there is carnivorous plants. Carnivorous is basically your insectivorous plants, so they also don't have such kind of feature. So that's why the correct answer is option A over here. Now, next is question number 85. Question number 85 says that plants having little or no secondary growth, you can say, they are grasses, deciduous angiosperms, cycads, and conifers. Now, let me tell you that secondary growth is a characteristic feature of those categories of plants which are monocots. All right, because secondary growth is not found in case of all categories of plant species. It is only found in case of the dicot plant species. All right, of the, and that too of the angiosperms I'm talking about it. All right, so definitely if you just look into the different options that has been given, grasses are the monocots over here. So grass are the monocots. So since grasses are the monocot plant and they do not possess secondary growth. So they usually do not have secondary growth, or they do not possess secondary growth. And other categories, if you will see, so this is a dicot plant species. Anything which is deciduous, this is a dicot category. Then this is a gymnospermic species, and this is also a gymnosperm. So this is also gymnosperm, and this is also gymnosperm. All right, so that is the case. So they do not, they usually have the presence, this is a gymnospermic species where the secondary growth is has not been reported, but there are some species that can show it also. So that's why we cannot consider these options to be the correct one. But grasses is the option in which we have studied regarding the secondary growth statement as well. All right, so that's why the correct option is your option A over here. Now, question number 86. Question number 86 says that the Casparian strips occurs in. Okay, so this question has been asked from the chapter of your transportation in plants of standard 11, and it is found in epidermis or pericycle or endodermis or cortex. Now, let me tell you that Casparian strips are those suberized strips, which are usually made of wax. All right, so we can say that Casparian strips are suberized strips. Suberized basically means that they are usually made of suberin and wax. So these are suberized strips, usually made of suberin and wax. And anything which is made of suberin and wax, so definitely that would be impermeable to water. So that is impermeable to water. So that means it won't allow the presence of Casparian strips, won't allow the passage of water molecules to pass through. So that is impermeable to water. So this is usually impermeable to water. All right. So, and it is found in, this is a characteristic feature which is found in endodermis. So it is found in the endodermis tissue of the plant body. So this basically looks like this. Like for example, if this is an endodermis, and this is a pericycle, and here is the cortex part. So it has the presence of Casparian strips like this. All right, so this is the strip which is present over here. So this is Caspasian strip. And this is endodermis. And because it is impermeable to water, so that's why the movement of water becomes symplastic, and then it becomes apoplastic like this in case of the transportation pathway. So that's why. All right. So the correct answer is option C over here. Now, question number 87. Question number 87 says that secondary xylem and phloem in the dicot stem, they are produced by. All right. Now let me tell you that this question has been asked from the chapter of your anatomy of flowering plants of standard 11. And the process of production of secondary xylem and secondary phloem in case of the dicot stem is a process of secondary growth. If the secondary xylem and phloem is produced, then this definitely indicates that secondary growth has occurred. And secondary growth is only possible because of the secondary tissues found in the plant body, and those secondary tissues are vascular ganglia. All right, so definitely we can say apical meristems won't be involved because this will promote for primary growth. So definitely this is not the option. And if you just look into this that has been given, that is secondary xylem and phloem is produced as a result of secondary growth. And definitely the secondary growth occurs because of secondary tissues, which is cambium. Secondary growth occurs due to secondary tissues like cambium. And if you have to form the secondary xylem and phloem, these are vascular tissues. So for the formation of xylem and phloem, definitely the vascular cambium is formed. That is, vascular cambium is involved in forming the secondary xylem and secondary phloem. So definitely this is the correct answer. Axillary meristem, again, this is also a meristem which will contribute in primary growth. So this is also not the correct option. Then there is phallogen. Again, this is also not the correct one because this is a category of cork. This will belong to cork category. So that's why this is also wrong. So that's why the correct answer is option B over here. Vascular cambium. Now, question number 88. Question number 88 says that you have to select the wrong statements regarding the following statement that has been given. So cell wall is present in the members of fungi and plantae. So this is again a true statement. This is a correct statement because if you just look into the cell wall, so cell wall of fungi that is made of chitin. So this is usually made of chitin, and the cell wall of plants that is made of cellulose. So this is a correct statement. Then mushrooms belongs to Basidiomycetes. Again, this is also a correct statement. This is also correct because mushrooms are the edible categories or edible food of Basidiomycetes. So that's why. So mushrooms are edibles of Basidiomycetes. All right, so this is also correct. Then the third says that mitochondria are the powerhouse of the cell in all kingdoms except Monera. All right, so this is also the correct statement because except the prokaryotes, the mitochondria is found in all kingdoms. That means it is found in Protista also, fungi also, plantae also, and Animalia also. So this is also true. That means this is also correct, you can say. So mitochondria is found in all kingdoms. That is Protista, fungi, plantae, Animalia, except Monera, that includes bacterial species. All right. And the last is that pseudopodia are the locomotory and feeding structures in sporozoans. Again, this is an incorrect statement because pseudopodia is known as false feet, and these are the locomotory and feeding structures in amoeba. That means amoeboid protozoans. They are not the category of the sporozoans. All right. So you can say that pseudopodia, which is also known as false feet, is found in amoeboid protozoans, not in the sporozoans. All right, so that's why the incorrect statement is this one. And since we have to find out the wrong statement, so that's why the correct answer is option D over here. Now, question number 89. Question number 89 says that which of the following statement is correct? Ovules are not enclosed by ovary wall in gymnosperms. Selaginella is heterosporous, while Salvinia is homosporous. Stems are usually unbranched in both Cycas and Cedrus. Horsetails are gymnosperms. Now, this question has been asked from the chapter of your plant kingdom of standard 11th. And let us just find out which is the correct statement. So, ovules are not enclosed by ovary wall in gymnosperms. So, this is again correct. This is a correct statement because ovules are naked. All right. So ovules are naked in gymnosperms. And since they are naked in gymnosperms, so that means this indicates that the ovary is absent. That means its covering ovary is absent. So that is ovary wall is absent. This is a correct statement. Now, Selaginella is heterosporous and Salvinia is considered to be as homosporous. Basically, this if you just consider the options that has been given, so both of them, Selaginella and Salvinia, both of them are considered to be as heterosporous. So Selaginella and Salvinia, and these are pteridophytic species. So Selaginella and Salvinia both are heterosporous. Both are. That means they will be producing two types of spores. One is the smaller microspores and the other is the larger megaspores, heterosporous. Now, the third is stems are usually unbranched in both Cycas and Cedrus. Now, stems are unbranched in Cycas, but it is branched in Cedrus and Pinus. So that's why we can say, so stems are unbranched in Cycas, but branched in cedrus. But it is branched in cedrus. So then this is also wrong. Horse tails are gymnosperm. Again, this is also wrong because horse tails are pteridophytes. These are not gymnosperms. So horse tails are pteridophytes. They are one of the categories of pteridophytes because it includes horse tails and ferns. All right. So that's why the correct one is option A over here. Now, question number 90. So question number 90 says that sweet potato is a modified stem, adventitious root, rhizome, and taproot. Now this question has been asked from the chapter of your morphology of flowering plant of standard 11th. And let me tell you that sweet potato is a category of modified adventitious root. All right. So sweet potato is a modified adventitious roots. Students often get confused with potato because potato is a modified stem. It's not a root, but sweet potato is a modified adventitious root whose structure has been modified for the purpose of storing food inside them. You know, it's a modified adventitious root for storing food. All right, so that is the case. While if you just, so the correct answer is this one only. While stem is a category, modified stem is a category of potato. All right, so this is the case. And if you just look into the rhizome, rhizome is a category, rhizome is also a category of modified stem. So this is showing also a category of modified stem. All right, so this is also wrong, and this one is also wrong. While on the other hand, taproot is one of the categories of root. All right, that results into, that is found in dicot plants. So this is found in dicot plants and is the elongation of radicle. So this is basically produced from the radicle. All right, so that is the case. That means it basically helps in the contribution of primary. Clear? So that's why the correct answer is option B over here. So this is all about the solution of your NEET 2018 question paper of your biology, and so we have successfully finished it and completed it. I hope it was readily understandable by all of you, and by this way we are going to study this and understand the questions in an appropriate way. So that's all for right now, and keep learning and keep scoring high. We will also be we will also be coming up with all different kinds of other kinds of video solutions as well that will be helpful in preparation of your examination. And for any doubt related queries regarding to the papers or any kind of solutions, you can join our Telegram channel that is given in the description box or the description section of the link. So that's all for right now. Thank you very much.